typical neuron in the central nervous system receives input from thousands of synapses. Integration of all of this input involves summation of the postsynaptic potentials in that postsynaptic neuron. We talked about summation, remember, when we were talking about adding together all of the depolarization of those graded potentials. So the greater the summation of the excitatory postsynaptic potentials, the greater the chance that threshold will be reached. So let's look at this image. There are two types of summation, spatial summation and temporal summation. So in the first image, A, when a presynaptic neuron 1 and presynaptic neuron 2 separately cause excitatory postsynaptic potentials in postsynaptic neuron number 3, the threshold level is not reached in neuron number 3. Spatial summation occurs when neurons 1 and 2 act simultaneously on neuron 3 and their excitatory potential sum to reach the threshold level. Then that triggers a nerve impulse or an action potential. Inside B, temporal summation occurs when stimuli are applied to the same axon in rapid succession. Okay, so it's causing overlapping excitatory postsynaptic potentials that sum. So when depolarization reaches the threshold level, a nerve impulse is fired. So during summation, it's taking more than one stimulus to cause that firing of an action potential. So as I mentioned in the previous slide, a neuron in the central nervous system receives a lot of input from many different presynaptic cells. So it's the net summation of excitatory postsynaptic potentials and inhibitory postsynaptic potentials that is going to determine whether or not that cell is depolarized to threshold. If threshold depolarization is reached, then an action potential will occur at the trigger zone here of the axon hillock, and an action potential will be propagated down the axon. If a neurotransmitter is going to stop acting on that postsynaptic cell, then it has to be removed from that synaptic cleft. Sometimes that happens simply because the neurotransmitter molecules diffuse away from the synaptic cleft. Once it's out of the synaptic cleft, it's basically out of the reach of the receptors. It can no longer exert an effect on that postsynaptic cell. Other times, certain neurotransmitters are degraded or inactivated by enzymes. For instance, when we looked at muscle contraction, we saw acetylcholine was the neurotransmitter released into the synaptic cleft that caused the muscle cell to contract. Now, one of the ways that the muscle was to relax was to remove that acetylcholine from the cleft, and it was the enzyme acetylcholine esterase that did that. Now remember, there was a lot more to muscle relaxation than that, and it had to do with the ATP and the calcium, but the enzyme acetylcholine esterase is what was responsible for removing that acetylcholine. So when the acetylcholine was no longer bound to the receptors, those sodium gates were not being triggered to open, and the postsynaptic cell was not being stimulated to depolarize. And then there is an action known as uptake or reuptake. So many neurotransmitters are actively transported back into the presynaptic neuron that released them. That action is known as reuptake. Certain drugs known as SSRIs, selective serotonin reuptake inhibitors, prevent serotonin, which is a neurotransmitter, from being taken back up into the cell. Other times, they are transported into neighboring neuroglial cells in an action called uptake. So once they're into the neuroglial cells, they can be destroyed. So we've got to get those neurotransmitters out of that synapse if they are to stop acting on the cell. Most cells make and respond to more than one neurotransmitter. There are about 100 substances are either known or suspected to be neurotransmitters. Some of these neurotransmitters bind to their receptors and act quickly to open or close ion channels in the membrane. So we say that those have a direct mechanism of action. Others act more slowly, and they use a second messenger system to influence chemical reactions inside the cell. So those take longer to act and tend to have a longer action. Those have an indirect mechanism of action, and of course the function of neurotransmitters can be either to excite or inhibit the postsynaptic cell. We also classify neurotransmitters based on their chemical structure, so what they're made of, how they're put together chemically. We'll take a brief look at some of the categories of neurotransmitters, including the biogenic amines or amines, which are modified amino acids. Some of the catecholamines you've probably heard of, as well as the indolamines, the dopamine, norepinephrine, and epinephrine being the catecholamines. Catecholamines are structurally different from the indolamines, and they have different actions. So the epinephrine and the norepinephrine are involved in arousal or waking from a deep sleep, dreaming, and regulating mood. The indolamines, serotonin and histamine, histamine, of course, is involved in the inflammatory response, and serotonin is thought to be involved in sensory perception, temperature regulation, control of mood and appetite, and helping us to get to sleep at night. So those SSRIs, selective serotonin reuptake inhibitors, can keep their serotonin in the synapse so that it continues to stimulate those postsynaptic cells and therefore increase our mood. These biogenic amines are said to be involved in, of course, emotional behavior and the biological clock, especially as associated with your circadian rhythm and the sleep-wake cycle. Some hormonal imbalances, some imbalances associated with mental illness, such as schizophrenia, are related to dopamine. So dopamine is going to be active during emotional responses and addictive behaviors and pleasurable experiences. I'm sorry, my tongue just got all tied up there. So let's start that again. Dopamine receptors are active during emotional responses, addictive behaviors, and pleasurable experiences. They also help to regulate skeletal muscle tone and some of the contraction of skeletal muscles. So the muscular stiffness that occurs as part of Parkinson's disease is due to the degeneration of neurons that release the dopamine. Also, there's one form of schizophrenia that's due to an excessive accumulation of dopamine. Some of the hallucinatory drugs, such as LSD, act on these same receptors that are bound by some of the biogenic amines as well. Peptides are short-chain amino acids. Some of them include neurotransmitters that help to mediate pain signals, such as substance P, so it enhances the perception of pain. Endorphins are our natural opiates, so they inhibit pain by blocking the release of that substance P. They were also thought to have a role in memory and learning, sexual activity, and control of body temperature, as well as mental illness. And gut-related neurotransmitter peptides are released by non-neuronal tissue in the gut, such as somatostatin and cholecystokinin, that help to regulate how full you feel and when to stop eating. ATP is one of the novel messengers that has a role as a neurotransmitter. So ATP is an excitatory neurotransmitter in the central and peripheral nervous system. A lot of the synaptic vesicles that contain ATP also contain another neurotransmitter that's released at the same time. Nitric oxide is a toxic gas. We don't store this as a neurotransmitter, but it's synthesized on demand and can be a very powerful vasodilator. Carbon monoxide, very similar mechanism to the nitric oxide, but it works on different areas of the brain. And next we'll move into neuronal circuits. Before we move on to neuronal circuits, I realized I wanted to talk to you a little bit about some of the things that can modify the effect of neurotransmitters, because this is important clinically. So there are substances naturally present in the body as well as drugs and toxins that can modify the effects of neurotransmitters. So neurotransmitter synthesis, or the making of neurotransmitters, can be stimulated or inhibited. For instance, patients with Parkinson's disease can receive benefit from the drug L-DOPA because it's a precursor of dopamine. For a limited period of time, taking L-DOPA can boost dopamine production in certain areas. Remember, dopamine is going to help regulate the skeletal muscle contraction. Neurotransmitter release can be enhanced or blocked. Amphetamines promote the release of dopamine and norepinephrine. Botulism can cause paralysis by blocking the release of acetylcholine. So if you don't release acetylcholine, then the muscles can't contract and it causes paralysis. Neurotransmitter receptors can be activated or blocked. An agent that binds to a receptor and enhances or mimics the effect of a neurotransmitter is an agonist. So there is a drug that works as an agonist of epinephrine and norepinephrine. So it can be used to dilate the airways during an asthma attack. Another drug binds to and blocks neuroreceptors for dopamine and serotonin, such as an antagonistic drug, Zyprexa. So it's prescribed to treat schizophrenia. Neurotransmitter removal can be stimulated or inhibited, as we talked about with the SSRIs. For example, cocaine produces euphoria, very intense pleasurable feelings, by blocking transporters that reuptake dopamine. So this action allows dopamine to stay in the synaptic clefts longer and produce that excessive stimulation of certain brain regions that are associated with feeling good. Okay, now we can stop with neurotransmitters and move on to the next topic.